Hello, my name is Brett Arenz. I am the director of the Plant Disease Clinic, which is based on the St. Paul campus just across the woods over there in Stakeman Hall. Raise your hand if you've ever brought a sample to the clinic. Okay, about half the people. All right. So today what I want to briefly talk about is some of the tree disease issues we've been seeing in 2023. Some, things that we see kind of every year, and some that are kind of interesting to this year in particular. And then also I will quickly mention a little bit about the Plant Disease Clinic, tips for sample submission if you're interested in using our services in the future. So first of all, what is the Plant Disease Clinic? So this is sort of what it looks like from Gortner Avenue there. So it's a service lab. We do have fees. They range from about $50 to maybe just over $100 for more time-consuming molecular testing, usually for things like viruses and things like that require a lot of bench time. And we provide diagnostic assessment of plant health problems. So from last year, we had just over about 2,000 samples, which is pretty average for us. And it represented over 170 different plant host species. So quite a diversity of different types of samples we get. Obviously, from your perspective, what you're probably most interested in, the type of trees and ornamentals and things like that. We also get a lot of field crops, corn, soybean, obviously being Minnesota, things like that. So a lot of diversity there. I just want to point out that about a third of our diagnoses, if not more, depending on the year, are actually no pathogen found. And what might be going on there are a variety of things. It could be nutrient issues, different sort of insect problems. Oftentimes the insects people can't see, like spider mites and wood-boring insects, things like that. Environmental damage, and then, of course, herbicide, chemical damage. So we don't do nutrient analysis in the Plant Disease Clinic. They do that at the soil testing lab, which is another service lab just south of here. So we do oftentimes bring samples back and forth if the clients are interested in that kind of thing, but it is a totally separate lab that does that. And then also we provide some degree of commercial sort of contract research support. Oftentimes this is maybe companies that are trying to develop some sort of potential pest control product or biocontrol product. They don't have their own microbiological lab and they want to run some tests. We can oftentimes work out an arrangement to do some of that kind of thing. So I thought what we'd do, shifting to some of the things that we've been noticing in the clinic this year, I thought I could show a picture and then see what you guys thought. So kind of a little bit of a more interactive kind of little plant disease quiz here. Sound good? Okay. All right, so what do you think we got going on here?
Drought stress.
Drought stress, yeah, I tried to fool you by taking the picture on a rainy day, right? But some things to look for, of course, marginal necrosis, indicating that the plant is having difficulty meeting its water needs, right? And I took this picture, or this is, in this case, a kind of a common scenario is you see portions of trees that are like hanging over roads and streets, pavements, things like that. And that's a really classic kind of scenario for drought and heat stress. A lot of different things going on here. You have the heat coming off the pavement. You have underneath the tree on the right side there, you can see there's a road underneath that portion of the tree. So you could, that part of the tree, its root system isn't going to get as much water. So even on a year, of course, where we have adequate water, you'll still see this happening really classically on trees that are in that kind of scenario, right? And of course, we've been in a drought situation in the last few years. This was a couple of years ago, the drought map in Minnesota, 2021, when it was quite severe. That was sort of the peak of the sort of recent drought. And of course, we have gotten, some more rain more recently, right? But this year in particular, we did go through quite a stretch with no rain since since May. And plants are going to react differently to drought stress, of course. Things like people's lawns and things will react to short-term drought stress and also recover from having more frequent rains. But trees, of course, there's going to be quite a lag in when they start showing signs of drought stress and also the time it takes to recover. So even though we might be able to get back our, some of our water table if we get some more rain this year, the stress they still show from the earlier period is still going to be definitely present. Okay, how about this one?
Oak wilt.
Oak wilt, yeah, is what I was going for here. And this is, of course, one of the most common types of samples we get in the clinic. And I should say that the testing request is actually one of the most common. We get probably 300 to 400 different oak samples per year. People wanting us to test for oak wilt, and there's a number of different reasons for that. Of course, it's a very severe disease. It should be managed if it is present, and it's really expensive to have to deal with treating the trees or removing the trees after they've died. So the amount of cost that we charge for testing, $65 or $85, depending on the species of the oak, is pretty minimal compared to that. So there's kind of a perfect storm of why we get a lot of this sort of sample. And the scenario I'm presenting here is essentially a situation where it's just a different angle here. You can see in this case, we have 3 different trees, right? The one in the middle there is obviously dead, okay, closest to the house. And it's sort of a really classic worst case scenario for oak wilt on a red oak, where you have it coming in, killing a tree. Tree is not removed. It's a source of inoculum for the neighboring trees, probably spreading simply by the root grafts to those neighboring trees. And if you have a scenario where you have especially red oaks growing next to each other and there's no management and one of them gets infected, it's sort of a car accident that you can't sort of look away from. So yeah, a lot of concern about oak wilt, and rightly so, because it's such a severe disease. Just a quick kind of recap about oak wilt. I'm sure a few of you folks know about this stuff, but just to kind of review. It affects red oaks more severely. They can die in a matter of weeks after being infected. Whites and burrs can live quite a bit longer, okay? And you could even say they can, in some cases, kind of compartmentalize it to a certain extent, right? Depending on what other stresses are going on. And you can even potentially treat them with a fungicide to sort of slow down the progression of the disease. With red oaks, of course, once they're infected, there's no possibility of treatment. You're really just looking at a removal scenario there. Of course, the main reason we want people to be aware about oak wilt is it's spread by these bark beetles that are attracted to wounds. Okay? And so when you're pruning an oak tree during the period when these beetles are active, that is when you're presenting a really risky situation. We advise simply, sort of no oak pruning during the sort of risk period for oak wilt, which you can check. Okay, there is an extension website where you can check there to see if in Minnesota we're in the active phase. And essentially, the safe period is from about November to March. The dormant period for the oaks, that's when oak pruning is recommended. The really dangerous period is sort of early on in the summer when they're sort of leafing out and the oaks are producing kind of that sort of early wood. And that's the really most vulnerable time. Now we're kind of trailing off into kind of an intermediate period where they're not quite as much at risk, but still pruning is not advised unless it's totally necessary, like an emergency situation. And if you do prune or you do collect samples for oak wilt, you want to make sure to treat the branch stubs with some sort of shellac or sort of latex paint so that the beetles are not going to be attracted to that fresh wound. And you can see there on a map the sort of distribution of oak wilt that the DNR has been kind of tracking in the last few years. And this was a suggestion, two-line chestnut borer has also become a pretty significant issue. And the symptoms of two-line chestnut borer can really closely mimic oak wilt. And so it's very difficult to tell in a lot of cases if an oak is affected by oak wilt or two-line chestnut borer. And so that's something we've seen really commonly in the clinic for samples that are submitted for oak wilt testing, okay? Especially on the whites and burrs, which are a little bit less susceptible to oak wilt. So two-line chestnut borer, these are going to be typically in trees that are under stress by all kinds of different potential conditions. Of course, drought stress potentially being one of those things. So you could definitely argue that this year has set up some oaks for more two-line chestnut borer damage. One thing to look for, if you do get the chance, is if you do look underneath the wood, you can see some definite clues about what might be going on here. So two-line chestnut borer, the discoloration you see under the bark will be sort of tunnel wound associated discoloration, oftentimes sort of meandering, kind of maze-like, sharp borders between the healthy wood and the discolored wood. Whereas oak wilt on the right, it's more streaky, it's more diffuse, more kind of gray. There's not a sharp line between the discoloration and the healthy wood. So that's definitely a helpful thing to look for if you're trying to make the distinction in the field. Okay, how about this one? Got a bur oak here. Leaf blister, right, good. Someone got it right away. So Tephrinia leaf blister, this is sort of on the left here is kind of what it looks like now, late season. On the right is what it maybe would look like earlier in the season, which is a little bit more distinctive, obviously. Now, of course, that tissue has sort of started to become necrotic, and it's a little bit hard to tell from some of the other oak issues. But kind of an odd one because it's been a dry year. We have not seen a lot of the kind of typical foliar issues we typically see with oak, like oak anthracnose, tubacia leaf spot, even bob. We're not quite sure how bad a bob year it's going to be yet. That's kind of a late season disease. But interestingly, Tephrinia leaf blister has seemingly become, or at least this year is especially notable. And it's a fungal disease. It likes wet conditions. So that's kind of odd since we had this sort of moisture stress. So as near as we can figure out, it probably was maybe a key period of time when the oak leaves were sort of emerging. And during that period of time, we did get some adequate moisture, and so the conditions were apparently okay for them to become infected with Tephrinia, even though after that time, it's been fairly dry. Okay, so kind of an interesting one, a foliar disease we didn't really expect with the dry year. Not nearly as severe as oak wilt. Certainly, a two-line chestnut borer would be more serious as well. Tephrinia is going to be more localized on the leaves. Okay, and so oaks can oftentimes, compensate for the damage. They can still get a lot of photosynthetic action, even if some of their leaves are looking a little tattered. And so really not as much of a concern as some of these other diseases. Okay, how about this one? I think we have like an Austrian pine here. Diplodia, yeah. So Diplodia tip blight is a very common disease for us to get in the clinic. I would say the majority of the pines that are submitted to us, this is what they have. Especially these non-native pines like the Austrian Scotch pines, Mugo pines, but also red pines are prone to Diplodia tip blight. You don't see it so much in something like a white pine though. Okay, so Diplodia tip blight is a really interesting one. It does affect the new growth. That's where you see the dieback. And a classic scenario and a really distinctive appearance is when you get dead tips and really short dead needles, which tells you that it killed it while those needles were growing. Okay, there's very few other pathogens that have that kind of appearance, right? And also, it's a latent pathogen, so infection can occur in a previous year, but you get no symptoms until the tree becomes stressed, and then it starts to express sort of Diplodia tip back. So a year like this year, where you're getting a lot of drought stress, might be actually a year where you see more Diplodia, just because the pathogen's already there, but it's really when the tree becomes stressed, that's when you're going to get the browning. But yeah, stress in older trees, most vulnerable. All right, how about this one? This is a needle cast of spruce. Okay, so classic symptoms there would be that it's the older needles, the inner needles on the lower part of the tree that are going to be affected first. That it's going to basically cause them to drop off the tree. And the tree that it's going to look like, it's eventually like a lollipop where it's just like the new growth at the top that you're going to see. But it's sort of like it's casting off its needles from the inside out. Really common sample for us to get on spruce. I should say that we get a lot of different issues on spruce, like cytospora, like insect issues, spider mites, spruce bud scale, all kinds of different things. So it's oftentimes a variety of different pests that are causing really nasty looking spruce, but this is definitely one of the more common ones. Stigmina needle cast, very similar disease. For whatever reason, we're not quite sure why, it has sort of replaced Rhizosphaera needle cast, but the symptoms are essentially the same. And it's really only underneath the microscope that you can tell it apart. So there's a little bit of difference in susceptibility. Something like a Colorado blue is typically most susceptible. Norway spruce is considered most resistant. I have seen it on Norway spruce, so they're not immune by any means. Increasing air movement, not having irrigation on the needles themselves, like sprinkler irrigation can really make it worse, that kind of thing. Making sure that the trees are not overlapping with each other. Oftentimes people plant spruce when they're young, they're too close together, and then they become crowded, you know. And fungicide application, if it is going to be used at like that time when the needles are actively growing in the early part of the spring and summer, okay, or late spring, early summer, because it's a protective. It doesn't get inside the tree, it protects the new growth. Okay, how about this one? Lilac we have here. It's kind of a newer disease, or newer in the news anyway. You're right, yeah, so this is an interesting one. Lilac leaf blight, Pseudosarcospora or Septoria. There's a little bit of a dispute about what name it should actually have. It's essentially the same disease. This is a really interesting one. It's not a new disease. It's been documented in Minnesota for several decades now, I believe. But for whatever reason, in the last 2 or 3 years, it has become more severe and much more noticeable. We've gotten a lot of calls about it. The Star Tribune did an article about it a couple of years ago because of all the interest. A late summer foliar disease. Okay, so you don't really see it until about August or even September. It really kind of depends on the year and what's going on with rainfall and things like that. Have not seen a lot of it yet this year. That being said, this was my dad's tree up in Bemidji. Okay, totally just torched, and that was a couple of weeks ago. The lilac right next to it looked fine. Okay, so not sure why that one was more affected than the others, but I have not seen it, for example, in my own neighborhood yet down in the cities. This is sort of what it ends up looking like. You get kind of patchy necrosis on the leaves. So not like that marginal kind of drought stress I was showing you before. Foliar, so it's only in the leaves. The trees can tolerate to quite a large extent because it is late, so they're doing a lot of the photosynthesis before they start to lose photosynthetic area late in the season. It's more alarming, I think, for people than it is actually serious for the health of the tree. But people have lilacs because they like the way they look, and if they start to look ratty, then it's a problem, right? So it's been kind of an interesting one. Still don't know a lot about what the best way of actually managing it is because it is sort of so recently emerging, I should say. Oh. Yeah, so there's some observations from the last 2 years. Did see it sort of mid-July in 2020, and kind of a rainy year, and then later on in the year in more sort of drier years. Okay, so just to kind of wrap up here, I just wanted to give a quick few tips about submission to the clinic if you're interested in using our services. So oak wilt submission, and any submission in general, is really only as accurate as the quality of the sample. So collecting a good quality sample is really critical. And that's why we recommend people submit living branches. So branches that are showing wilted leaves but have not become brown and totally like firewood kind of dead, right? That's going to be where we're most successful in actually getting the pathogen out. So leaves that emerged this year and now are starting to wilt, that's the branch that is ideal for sort of collecting. 1/2 inch to 1 inch in diameter, maybe 2 to 3 inches in diameter is really sort of what we're looking for. We can't do much with twigs. It's very hard to get oak wilt out of that. So collecting samples during the active wilting season is usually the recommendation. That being said, when you're collecting them, you want to make sure you're treating the branch stubs immediately afterwards, right? Also, just to mention, oak wilt is not uniformly distributed through the tree. In a lot of cases, you might have one branch with it in it and one branch without, especially in something like a white or a burr. And so if somebody just randomly collects a branch, maybe in the winter when they can't see the leaves, and they end up picking the wrong branch, then they might get a false negative result. That's why it's really important to primarily do it during the active wilting season. And then keeping the sample cool, submitting it soon after collecting is useful. Don't try to leave it in a vehicle for weeks on end. After it dries out, it can be really hard to work with. Okay, so just a little bit more about the clinic. Again, it's Stakeman Hall, just across the woods over there. The university has recently kind of changed their security measures. Most of the buildings are going to be locked to the public, but we have been able to successfully lobby to keep the door of Stakeman Hall open. during our business hours, so that that eastern facing stakeman door will be open from like 8 to 5 during the week to drop off samples. Living plant material, as I mentioned, is typically the best, so showing signs of the disease, but not yet totally dead. It's hard to kind of do autopsies with some of these diseases because they become kind of quickly colonized by other microbes. We can take samples hand delivery or in the mail. If you're interested in sending in the mail, we recommend wrapping the roots in plastic and the above ground plant material in newspaper. And we're talking about like a smaller plant, obviously, in this case, not like a whole tree. What happens there is that it keeps the plant from drying out because the roots stay moist, but the above ground part doesn't become totally covered in mold like it would if it was just in a plastic bag in the mail for 2 or 3 days. And then as much information as you can provide in the submission form, which we have online, or we also have them in the clinic, is always good. Call, email with questions, and then I just have our email up there. And then just some acknowledgments. If you come into the clinic, these are a couple individuals you might see, Jennifer or Grace. So, and some people that provide some diagnostic help for us. So thank you very much. Anybody have any quick questions? What I've been seeing mainly this year is spider mites. I should probably have mentioned, I didn't really have time to get into this, but some of these insect issues can become worse when you have drier conditions, because oftentimes the natural control of their populations are different fungi that actually attack the insects or mites. And so when you have drier conditions like we've had this year, things like spider mites and aphids and things like that can become a larger problem. So I would say this year it's mainly been spider mites I've seen on fur, causing kind of a rusty looking appearance on the needles. And typically they're too small to be seen without a microscope. So that's why oftentimes people miss them and they don't know it's a spider mite situation. Not necessarily. Some diseases are kind of associated with that leaf retention, like bur oak blight, for example. I have not heard that for two-line chestnut borer especially. So yeah, but you could certainly check that because you'll see the signs of tunneling even in dead branches from two-line. So that can be pretty easy to check.